So in this biology session, we're going to be going over the end of chapter 12, section 3, about DNA. And we know that every cell in the body has a copy of DNA. It's got the DNA for the whole entire organism, and that DNA is passed from parents to offspring in the gamete cells. The process of copying the DNA is called replication, and that's what we're going to be focusing on. Okay, we're going to figure out how um, that DNA is copied, because to be in every cell and to be passed down, it has to have copies of itself made. And that's a really complex process because DNA is huge and complex. So we're going to look at how that's done. So in DNA replication, enzymes untwist the DNA and unzip one strand from another, separating the base pairs. So the enzyme goes through and splits the DNA in half. So you have one strand on one side and one strand on the other. They've been separated. And enzymes are a type of protein, and they do a lot of different jobs in the cell. So we're going to talk a bit about other things that enzymes do. So once they're split apart, another enzyme called DNA polymerase, and that's a name that you should know, DNA polymerase, that attaches nucleotides with matching base pairs to each side, making two new strands. So we learned last lesson that A bonds to T and C bonds to G. So these DNA polymerases are going to come in and bind new nucleotides to the existing strand using that base pair. So, okay, we started with, oh, that should be a G. Or an A, I'm sorry. So, okay, I made a mistake. Your DNA polymerases don't usually make this mistake. Your T bonds to A, C bonds to G, T bonds to A bonds to T, G bonds to C, T bonds to A. So we've got a nice matching set of nucleotides, and they all bonded to there. And then on the other side, it's going to go in the opposite direction, but do the same thing. It's just going to bring in all the matching nucleotides and bond them up. And now we have two complete strands. So we started out with one strand of DNA, we split it in half, gave each half a matching strand, and now we have two complete strands. Okay, so it looks something like this if you're looking at a slightly bigger view of it. This is just stating the same thing another way. After the strand is split in half, so up here you can see we have like a whole normal strand of DNA, and here it's being split apart. Okay, so after it's split in half, each half is made into a whole strand of DNA by DNA polymerase. So you can see here's your DNA polymerase. I'm going to put a little P on there. And it has gone up through these bottom pieces and added the other half, and it's doing that here. So here you've just got kind of a naked half of DNA that doesn't have the other pairs added to it, but this DNA polymerase is going to go through and continue adding them. And so it's making a complete strand over here that's double-sided. And the same thing's happening on the left side here. So we have it split apart and it's just working in the opposite direction. It's going downward, but you had a DNA polymerase come in and fill in the other half. And down here again, you have a DNA polymerase that came in and is filling in the other half. Now this is just an animation that's going to show that happening. So we've got a DNA strand, a double strand of DNA, just normal DNA. So first it gets split apart by an enzyme called DNA helicase, and now another enzyme comes in and is adding the matching base pairs. So I have to do that manually. I'm pretending I'm the enzyme here. So you've got a C coming in to bond to that G, and A coming in to bond to that T. Oh, I need a T up here. And then we just keep going. A bonds to T, G bonds to C, C bonds to G, A bonds to T, and DNA polymerase does this a lot faster than I am, and you can't mess it up. If you try to bring in the wrong letter, if you try to bond a G to a T, you can see they don't match. They're not the same shape, and they're not going to bond. Okay, so in real life, they're just molecules that wouldn't bond together, so it doesn't get mixed up. A bonds to T. And as we keep going, you're going to see that the end result is going to be two completely identical strands of DNA. All right, so now we're all done with what's available. You can see what we have on the left and what we have on the right are two complete strands, and they're absolutely identical to each other. So when DNA is copied, it's turning from one strand of DNA into two complete strands that are absolutely identical to each other. They have the exact same base pairs. Okay, so some other details about it. To talk about telomeres in your book. So telomeres are kind of the end caps of the chromosome. So if you have a chromosome here, these pink areas would be the telomeres. They are made of DNA, but they don't code for any genes. They're just nonsense DNA. They're random letters that are repeated over and over that don't mean anything. They're just kind of protective caps, and they're interesting. They get shorter on every newly made copy of DNA, and that is a big factor in aging. So these telomeres getting shorter and shorter and shorter actually limits how many times each cell can divide, and that affects your entire body because as your cells age and they can't divide more and you get more and more aged cells, that's kind of what contributes to people aging. As you get older, your body kind of starts to deteriorate. And it has to do with a lot of things, but one thing that is in effect is these telomeres getting shorter. So they've been doing a lot of studies on this lately. One thing they found out years ago now, but that's still being researched a lot, is that there's an enzyme called telomerase, and it helps to reverse this whole shortening thing. So normally every time your DNA shortens, the telomeres or the DNA is copied, the telomeres get shorter and shorter. With telomerase, it makes them longer again, so they don't keep getting shorter. So telomerase is active in the cells of young individuals, and it happens in some cells that have to reproduce often, such as the stem cells, cells that make egg and sperm cells. But it's not active in most adult cells. So something interesting they figured out is that it is active in a lot of cancer cells, and the whole thing with cancer is they're cells that reproduce when they shouldn't. So like cancer can create tumors because they're just masses of cells that just keep reproducing over and over when they shouldn't be. So um, because telomeres are important in aging and they seem to be important in cancer, it's something that's being studied an awful lot. So I just wanted to go over this so you had some idea of what they're talking about. If you read a lot of news articles about genetics or science or cancer or aging, you're going to see about telomeres. So scientists are studying ways to fight the disease of cancer and some other diseases and kind of see if there's any way to combat aging to keep people more youthful by activating or deactivating this telomerase. Okay, so that's just important to know for this class and something that you should be aware of because you're going to see it a lot in the future if you read any science news articles. And then the chapter here ends with comparing replication in eukaryotes and replication in prokaryotes. So eukaryotes are what we've been looking at all along. Okay, this replication of DNA, we've been looking at prokaryote DNA basically, but it's pretty similar. So the only thing we didn't mention that it does tell you at the end is that it happens at several points on the chromosome at once. So to make it simple, we were just kind of looking at one area where your two strands of DNA split and new DNA was being added, but that's actually going to happen at several places at a time, which kind of helps it be more efficient. DNA is very, very long. It would take forever if there was just one DNA polymerase on each chromosome. It would take forever to divide. So this is just happening at quite a few locations along each chromosome. And then prokaryotes are a little bit different. Remember that prokaryotes are cells with no nucleus, and their DNA is different. They have one big circular chromosome instead of that long twisted strand of a chromosome. So their DNA is shaped different, but really replication is similar to eukaryotic cells. The two original strands split apart, and then matching strands are added. So it looks something like this, where your original splits off and those dots are showing where the matching ones are going to be added. So just like the eukaryotes, where we had kind of a left side and a right side and they split in half, and then we added more bases to each side, that's what's happening here. We have the inside and the outside, they split in half, and we're adding the new bases onto what's already there. So it's a really similar idea. It's just that it's in a circle and it only starts at one place at a time, whereas with the eukaryotic cells, we just saw that it does several places at a time. And that does bring us to the end of this chapter, but if you have any questions, don't hesitate to contact an instructor.